to Learn Classic Education. Today, we're going to discuss the gluconeogenesis topic, an important topic of biochemistry. Okay, this process is essential for maintaining blood glucose level, especially during fasting or prolonged exercises. Okay, it occurs primarily in liver and to lesser extent in kidneys also. So, let's dive into the topic. Okay. So, first we'll discuss what is the definition of gluconeogenesis. So, gluconeogenesis is the synthesis of a glucose from non-carbohydrate precursor, maybe like lactate, amino acid, in glycerol. Okay, so we can say gluconeogenesis is the reversal of glycolysis, right? Which primarily occurs in liver and kidney. Okay, what happened in this? Two pyruvate molecule turns into the glucose. We have discussed in the glycolysis where the glucose molecule turns into two pyruvate molecules, but here we know that gluconeogenesis is the reversible of glycolysis, in which two molecules of pyruvate will give us to one molecule of glucose. Okay, now, what are the importance of gluconeogenesis? As we discussed, it maintains the blood glucose level during fasting. It provides glucose to the brain, RBC, and other organ-dependent tissues. Then it clears lactate from aerobic metabolism, that is the Cori cycle that we'll discuss in the later parts. And also, it prevents hypoglycemia in prolonged fasting period. Okay. Now, what are the sites we discussed? That is the liver and kidney, and in the cellular compartments like mitochondria and cytoplasm. Okay. Now, some important enzymes of gluconeogenesis that includes pyruvate carboxylase, PepsiK, fructose 1,6 bisphosphate, and glucose 6 phosphate. Okay, so glucose 6 phosphate is an important enzyme that is seen in the liver and the kidney. So pyruvate carboxylase, PepsiK, fructose 1,6 bisphosphate, and glucose 6 phosphate. Okay, now moving to the steps involved in gluconeogenesis. So, first step includes conversion of pyruvate to phosphoenol pyruvate, right? PEP we can say. So, pyruvate with the help of uh, pyruvate carboxylate, it converts into oxaloacetate, and this oxaloacetate with the help of PepsiK convert into phosphoenol pyruvate. Okay, then this step bypasses pyruvate kinase of glycolysis. Okay, then we have second step in which the conversion of fructose 1,6 bisphosphate to fructose 6 phosphate by the enzyme fructose 1,6 bisphosphatase. Okay, this is important because it is a rate limiting step and a irreversible step. Then another step, the third step, is the conversion of 6 phosphate glucose 6 phosphate to free glucose molecule. Okay, it is also irregular step which is catalyzed by glucose 6 phosphate enzyme phosphatase enzyme. Okay, so total in this step 11 ATPs are used, right? So 11 ATPs are used. Now we're moving to the topic malate shuttle. So the malate shuttle is a process in which the oxaloacetate from the mitochondria is transported to the cytoplasm in gluconeogenesis. Okay, so in a nutshell, we can say that the malate aspartate shuttle is a biochemical process that transport NADH from the cytoplasm into the mitochondria. Okay, so this shuttle is essential because the mitochondrial inner membrane is impermeable to NADH. Okay, but NADH needs to enter the mitochondria for the ATP production in the oxidative phosphorylation. So that's why uh, we need to transport the oxaloacetate from the mitochondria to cytoplasm. Okay, so what are the steps involved in malate aspartate shuttle? So the first step is that cytoplasmic reduction of oxaloacetate to malate, because oxaloacetate cannot transport, or they cannot cross the mitochondrial membrane, so it is converted into cytoplasma malate. Okay, then the help of enzyme malate dehydrogenase. Okay, in the reaction includes oxaloacetate plus NADH plus hydrogen atom gives malate plus NAD plus. Okay, Then NADH donates electrons converting oxaloacetate into the malate. Okay. Then second step includes the transport of malate into the mitochondria. Okay. So then malate cross the inner mitochondrial membrane via the malate alpha ketoglutarate transporter. Okay. Then the third step includes the oxidation of malate to oxaloacetate in mitochondria. Okay, with the help of enzyme malate dehydrogenase. This is a mitochondrial malate dehydrogenase enzyme. Okay, the reaction includes malate plus NAD plus gives oxaloacetate plus NADH plus hydrogen atom. So now see, we have transported the oxaloacetate from the from the mitochondria to the cytoplasm. Okay, then NADH, which is inside the mitochondria, enters into the electron transport chain to generate ATP. Okay, now this oxaloacetate convert into aspartate with the help of enzyme aminotransferase. Okay, so aspartate is formed to allow transport back into the cytoplasm. Okay, now the transport of aspartate back into cytoplasm occurs with the aspartate glutamate transporter, right? So now we will discuss what is the use of malate shuttle. So the first use of malate shuttle is to transport oxaloacetate from mitochondria to cytoplasm, right? Then to transport two molecules of NADH from cytoplasm to mitochondria in aerobic glycolysis. Okay. Now, ATP generation is also a significance of malate aspartate shuttle, as NADH transferred into the mitochondria donates electron to electron transport chain, which produce 2.5 ATP per NADH. Okay. Also, the significance includes active in liver and heart and kidney, like organs with high energy demands utilize that shuttle extensively. They use a lot of this malate aspartate shuttle. Okay. See, now what is the difference between using the malate shuttle for both gluconeogenesis and glycolysis? See, gluconeogenesis and glycolysis both use malate shuttle, but in the case of glycolysis, it occurs anaerobic. In the case of anaerobic glycolysis, NADH is not enter into electron transport chain. Okay, and in the case of aerobic glycolysis, malate shuttle and glycerol phosphate shuttle is used. So now regulation of glycolysis and gluconeogenesis, both are reversible to each other as we have discussed, right? And they are regulated in such way, if one path is activated, other part should be inactive, obviously, right? And otherwise they will become waste or fertile, right? There is no use of doing that, so we will regulate only one path at a time. It should be a gluconeogenesis or glycolysis. So let's recall everything. Now, what is gluconeogenesis? So gluconeogenesis is a synthesis of glucose from non-carbohydrate precursor like amino acid, glycerol and lactate. And we also know that the gluconeogenesis is the reverse steps of glycolysis, right? Then what are the sites of gluconeogenesis? So mainly they occur in the liver, right? That is the primary site. And also it can occur in the kidney, right? When we are in prolonged fasting state. And cytoplasm and mitochondria, some step occurs in different compartments too, right? Now, uh, what are the important enzyme of gluconeogenesis? That is the pyruvate carboxylase, pepsi-k, fructose 1,6 bisphosphate, glucose 6 phosphatase, right? Now we have discussed the important steps that are involved in this. That includes pyruvate to phosphoenol pyruvate, fructose 1,6 bisphosphate to fructose 6 phosphate, which is the rate limiting step and irreversible step, and also glucose 6 phosphate to glucose, that is the irreversible step. And in this process 11 ATP used, right? We also discuss about the malate shuttle, right? Now this is all about from my side to gluconeogenesis, right? If you want to learn more about it, please book a class from Learn Class Education, right? Okay, bye, take care.